Cut to the chase, evidence-based. Pull up a chair, let's get this straight. Peptide buddy, he's your peptide buddy.
Hey everybody, in a recent episode of the Joe Rogan Experience, Rogan and another podcaster, comedian Tom Segura, took a 5-second break from talking about how much money they make to mention peptides. And I was hoping they'd get more into it because it's obvious Tom's on peptides. I know Rogan is too. It's been mentioned before, but it seems Rogan really had no interest in continuing the conversation on this podcast. But it's an interesting discussion because we have to guess what peptide Tom's referring to. So let's watch the clip real fast.
I've been doing that. I have a whole new sleep protocol I'm doing.
Oh, a sleep protocol.
With, like, I'm saying a bunch of peptides and yeah.
Ooh, I just ordered an Oura ring to track my sleep.
That's good. Do you have to try that pineal peptide?
No, I haven't. I heard about it. You were telling me it really ramped up your REM sleep.
Awesome.
Yeah?
Yeah.
Nice.
Yeah. Anything for more sleep I'm into, man.
I'll tell you what really affects
quality sleep.
No drinking.
Okay. So last time we talked about Pinealon, we revealed a very common misconception and addressed it. There are plenty of people who understandably think that Pinealon is derived from the pineal gland when that's not really the case. In fact, another peptide within the same class, Epitalon, is more directly related to the pineal gland than Pinealon itself, which I know is a bit confusing and maybe even surprising. And so I think the brief interaction between Rogan and Segura gives us a good segue into a greater conversation about these two peptides. So consider this a combined lecture on Pinealon and Epitalon. I covered Pinealon recently, but it's been a minute that I gave Epitalon the full focused video it deserves, and I know a lot of people are interested, so I wanted to get back to it. I will warn you, the beginning of this video, and actually most of it, is going to be quite dense, but I think it's necessary to distinguish between a couple popular ideas in the space, which come with them their own misconceptions. I'm confident, however, that by the end of this video, you'll be better able to describe what a Khavinson or bioregulator peptide is, what it's intended to do, and the differences between epithalamin, Epitalon, and Pinealon, and finally what we know about Epitalon and Pinealon in particular, and how engraved these popular ideas about their mechanisms and their uses, and really how much research supports their popular use. Epitalon is a synthetic tetrapeptide derived from the amino acid sequence of epithalamin, which in and of itself is an extract initially isolated from the bovine pineal gland. Its structure is alanine, glutamate, aspartate, and glycine. And as you'll see when doing your own research, a lot of the data that's publicly available has been detected from epithalamin, this precursor rather than Epitalon itself, which is its own limitation in our comprehension, but we'll get into that in a bit. Pinealon consists of fewer amino acids, only 3 in fact, which makes it a tripeptide, glutamate, aspartate, and arginine. And so you'll see it named for its structure as EDR, contrasting from Epitalon's 4 amino acid structure termed AEDG. Some overlap, which is clear, but at the core of these Khavinson or bioregulator peptides lies the idea that the actions of these small, tiny compounds are modulated by their specific structures. In other words, these compounds are isolated from specific sources, and as such, their synthetic replicas are thought to induce geroprotective and anti-aging properties of those sources themselves. It's why cardiogen is thought to functionally benefit the heart, prostogen the prostate, Epitalon the pineal gland, and ironically enough, Pinealon, not so much the pineal gland. Rather, Pinealon was founded when researching Cortexin, a synthetic peptide that's gained popularity in Russia due to its purported neuroprotective and neurotrophic effects. So we could say it's more derived from the brain cortex than it is the tiny sleep-regulating pineal gland, unlike Epitalon, which one could argue is derived from the pineal gland directly. The point of all these words is to emphasize that understanding the actions of Epitalon is unlikely to be achievable by looking at data on epithalamin alone, not only because they're different compounds with different names, but because their chemical structures are different. And after reading the work of the founder of these bioregulator peptides, Vladimir Khavinson, it would be presumptuous of us to assume they are the same when the discoverer himself highlights the precise roles of these peptides are structurally dependent. I will add that Khavinson's work reflects a lack of clarity on Epitalon's derivation. While it's more clear epithalamin is intended to replicate pineal gland activity, Epitalon is thought to probably reflect the embryonal origin of the pineal gland and the retina, so likely a component of ectoderm. In early stages of human development, different germ layers proliferate and unfold to give rise to different tissues and organs. This shared embryonic origin underlies the rationale for the design of bioregulator peptides like Epitalon, which are thought to target transcriptional machinery common to both the pineal gland and the retina, reflecting their ectodermal lineage and potentially explaining their overlapping biological effects. This interpretation aligns with Khavinson's peptide theory of aging, which posits that tissue-specific peptides can regulate gene expression and maintain organ function through mechanisms rooted in their developmental lineage. I think a good line to conclude this section of the video is to take a quote from Khavinson himself, that within this theory, aging is an evolutionary determined biological process of changes in gene expression resulting in impaired synthesis of regulatory and tissue-specific peptides in organs and tissues, which provokes their structural and functional changes and the development of diseases. And that's exactly what his bioregulator peptides are intended to do, directly confront genetic features of aging. Now, I should note, as I have in the past, that much of Khavinson's research, while prolific, lacks the kind of large-scale, independently replicated trials we'd typically expect in Western medical literature. That doesn't mean it's wrong, but it does mean we have to interpret claims about transcriptional regulation and tissue-specific rejuvenation cautiously, since they haven't really been acceptably proven in humans, and citations that claim it has are not really made available for us to fact-check. And when they are, they lead their own series of questions. So let's just jump back into where we were earlier. We've got 3 peptides on our docket: epithalamin, the pineal extract from animals; Epitalon, a synthetic version based on epithalamin sequence; and Pinealon, a different tripeptide EDR, more cortical than pineal in nature. Now, among the many claims that Epitalon fans favor are a few that stand out to me, which in my opinion lead many to the peptide, which has been considered a possible fountain of youth. First is the idea that it increases telomere length, which would support anti-aging and lifespan prolongation, at least in theory. That telomere length correlates in a way with youthfulness and disease protection. And the other is that it would prime the pineal gland to produce melatonin, which is also thought in some spaces to generate an anti-aging response and more directly target sleep. And it's about at this point that I realized the video isn't going to be as straightforward as I was initially hoping, because a lot of the rodent research that propelled the aforementioned ideas is from Khavinson's circle in the late 70s and early 80s, and so comprehensively blending these findings with the more current day reviews of the data is quite difficult, but I'm going to try my best, as I always do. And by the way, if you're still here with me and appreciate the effort of your peptide buddy, make sure to hammer that like and subscribe. Feel free to dislike if you don't like the video, but I do appreciate the time you take to watch this. I know we're almost at 10,000 subscribers, which I didn't realize was a possibility a year ago, and so if you could help me get there, would appreciate it significantly. Okay, so there exists a 2025 literature review that I think does a good job of synthesizing a lot of the data, not all of it, but a lot of it, which we can use to our advantage in crafting a well-rounded understanding of the elusive Epitalon. Now, the researchers adequately state in the beginning that the more researched compound is epithalamin, which we did mention in the beginning of this video as well, and it's important to point out. Now I'm going to list some of the pertinent findings from the collection of preclinical research on Epitalon, and then we can acknowledge as a whole really what this all means. In models of retinal degeneration, Epitalon appeared to help preserve retinal structure and visual function, and in some in vitro contexts it supported pigmented or neuroepithelial cells, though it's not clear whether it directly promotes proliferation. In rodent pineal light cells, Epitalon showed changes in certain laboratory-evaluated markers that would suggest an effect on melatonin synthesis. A similar preparation showed Epitalon may enhance features of lymphocyte maturation, hinting at an unclear relationship with the formation of cells involved in immunomodulation and immune response. In mice splenocytes, Epitalon seemed to increase expression of interleukin-2, IL-2, which could further hint at some sort of intricate relationship with the immune response. And research on the African clawed frog, or Xenopus laevis, pluripotent cells indicates that Epitalon exhibits a concentration-dependent biphasic effect on cell fate determination, meaning that different doses of Epitalon could potentially lead to various cell differentiation outcomes. This finding suggests that Epitalon may act through tightly regulated signaling pathways, where only specific concentration ranges are effective in promoting particular outcomes. Now, if you think we haven't gone deep enough, it's worth diving even deeper on a more detailed level on the data related to telomerase activity, because there is this theory that's growing more and more popular and becoming more investigated on the relationship between telomere length and aging in such a way where the length of the telomere is thought to mimic youthfulness and more youthful states. And as we age, telomeres shorten and signal more of a decline in aging functionality and growing susceptibility to diseases of aging. So in human fetal lung fibroblasts and the telomerase-positive cancer cell line used in research, Epitalon led to longer telomeres during a specific phase of cell growth. As a result, cells treated with Epitalon were able to keep dividing for more cycles than untreated cells, which is thought to be one way it might slow down cellular aging. Another study in lymphocytes extracted from elderly human participants compared to a control of cells taken from younger adults showed an increased number of acrocentric chromosomes per cell, which isn't particularly relevant. However, Epitalon treatment appeared to be related to dechromatization of chromosomes 1 and 9 in particular. Together, these findings indicate there may be a relationship between Epitalon administration and features of aging. Epitalon-induced deheterochromatization of regions of chromosomes 1 and 9 leads to what's essentially a loosening of chromatin structure, which could possibly reactivate previously silent genes and restore ribosomal gene activity and proliferative effects. An optimistic theory, but an idea here. This proposed chromatin remodeling is considered beneficial as it could theoretically counter the age-related repression of gene expression and support cellular synthetic processes, potentially improving the functional capacity of aged cells. Additionally, in blood lymphocytes, telomere length was found to be influenced in some young and middle-aged individuals, with some showing no significant changes, some showing increased telomere length, and some showing decreased telomere length. Overall, Epitalon was shown to induce the expression of telomerase enzyme components, telomerase activity, and telomere elongation by an average of 33.3% in human somatic cells, indicating that Epitalon could possibly modulate telomere biology in human lymphocytes, but with interindividual variability in response. Now, I know that was a lot, but the point to saying all of these words is that Epitalon's effect on telomerase is one proposed idea that leads a lot to the hype surrounding the peptide. However, if anything remains clear, it's that these effects are so unclear. When we get to the genetic level, things are indescribably intricate and precise, and the variability and heterogeneity of results emphasize that yes, there's some relationship between Epitalon and telomere length and telomerase function in some individuals and some cell types, but nowhere near appropriate to say what will happen to your body if you inject the peptide. What remains unknown is why individual responses are so variable. What's the long-term safety of telomerase activation if we're thinking optimistically? And do these cellular effects translate into meaningful clinical anti-aging benefits in humans? The precise molecular pathways and optimal dosing regimens also remain to be clarified, given the effects are so variable and inconsistent even in these controlled cell lines. So to recap, before we go even further, this is what we've got so far. Epitalon's shown some sort of preservative effect on retinal cells, possible involvement in melatonin synthesis, proposed immunomodulatory effects, and an unclear but interesting relationship with telomere function and the genetic patterns of aging. This is an adequate time to reinforce that a lot of Khavinson's research, where results of Epitalon are quoted in humans, such as that regarding visual function, are based on limited selectively available research. There's generally speaking, a lack of transparency, which doesn't equate to BS or non-existence. In my mind, however, I attribute that to differences in Eastern European research standards, but it's tough for me to draw firm conclusions in the absence of the kind of robust, well-elucidated protocols we'd expect in Western clinical research. The available studies often lack the methodological rigor, sample size, and transparency that are expected in contemporary clinical work, and independent replication is limited. At At the end of the day, despite my passion for going through all I can and synthesizing it for a few of you, I'm not the arbiter of what peptides are good and what peptides are bad, right? You see plenty of comments that say, just try it, bro, the results will speak for themselves, but that's not the purpose of this channel. If you want that, you can find the hype on most other channels that reference peptides. I treat my reviews as unbiased as I can. I don't cherry pick data, and I'm not affiliated with any compounders. I'm just a YouTuber practicing an honest man's work. And there's plenty we're not touching on. There's not enough time in the world, but the literature review I'm highlighting in my reference list is, in my opinion, one of the most comprehensive and well-constructed overviews available, and I suggest you look at it if you're so interested. I want to emphasize some other research that's been carried through Khavinson's literature some time now. This is where Epitalon showed some lifespan prolonging effects, up to 16% in the common fruit fly, and that effect was mainly seen in the older, more mature flies. But this effect doesn't appear to be universal, and a specific strain of mice didn't show any lifespan prolonging benefits from the peptide. Additionally, in her two new transgenic mice, Epitalon did reduce tumor burden, but at certain doses it may have been associated with reduced survival, perhaps due to increased frailty in these animals, suggesting that Epitalon's dose curve is highly intricate and not fully understood, which we really already knew. The broader picture here is that Epitalon cannot be assumed to be universally beneficial. While some studies show it can enhance features of anti-aging, others suggest that under certain conditions or in certain models, the effects may be neutral or even counterproductive. You'll see in some animal populations there's a beneficial effect, in some there's none, and in some there's the contrary. I will note that the positive outcomes too, in addition to the tumor suppression noted earlier, Epitalon's demonstrated antioxidant, anti-mutagenic, and cytoprotective effects in various models. In some studies, it reduced the incidence of certain tumors, such as leukemia, though it didn't always affect total tumor incidence across the board. The beneficial effects of lifespan are often observed at extremely low concentrations, especially in invertebrate models, and the dose-response relationship is complex, nonlinear, and not well understood. Other factors that may influence Epitalon's effects beyond dosing include the species being studied, the age of the organism, and even environmental conditions like light cycles or stress exposures. And just to be clear and highlight the obvious, none of this has been remotely or considerably explored in human lifespan studies, at least nothing that's made available. And on top of that, when we're talking about telomerase and elongation of telomeres, does this propose a cancer risk, since cancer cells grow and spread due to upregulated telomerase activity in many cases, and it's a pro-cancerous outcome. Although the idea of increasing telomerase expression is a big one in the anti-aging biohacking field. Epitalon's antioxidant effects seem to come primarily from its ability to upregulate key cellular defense enzymes, rather than directly scooping up free radicals. That's a little different from melatonin, which acts more like a direct scavenger. And while there's some speculation that Epitalon might interact with transition metals and help blunt oxidative chain reactions, that part isn't really particularly confirmed. Either way, these effects may contribute to its broader cytoprotective and geroprotective potentials. Additionally, as it turns out, Epitalon is a pretty stable tetrapeptide. Physicochemical studies suggest it forms what's known as a stable beta-turn structure, held together by internal salt bridges and hydrogen bonds. That compact conformation limits its flexibility, which might explain why it's been shown in simulations to bind specific DNA sequences found in the promoter for telomerase. The idea here is that the shape and sequence interaction could help reactivate certain genes, which is a cool theory, but we don't have a crystal structure or clear downstream effects, just some data that suggests involvement within genetic regulation. And that's where things also get a bit murky, because these kind of effects, structural binding, chromatin loosening, gene activation, they sound great on paper with regards to geroprotection, and when we're talking about them on YouTube, yeah, they sound pretty cool, but they're quite sensitive to pH, concentration, and the intracellular environment. So in other words, Epitalon has physical properties that support its proposed function, but no, it doesn't mean that it's quote-unquote unlocking anti-aging with a single injection. It's a peptide, tiny, fragile, and context-dependent, and whether that beta turn is doing anything useful in the body, it's still a big maybe. These are questions that all need answers, and we're working on this, microphysiological level, so hard questions to answer as is. Now, I know this video turned quite obviously into more of an Epitalon repackaged than a Pinealon comparison, which was in a way intentional, but let's switch gears a little bit and rehash Pinealon, a peptide we've covered much more recently than we had Epitalon. As we touched on earlier, Pinealon is really more of a cortical peptide, thought to protect neurons, prevent apoptosis, and improve cognitive functions like attention, memory, perceptual motor responses. Mechanistically, it's been tested in vitro in hippocampal and cerebellar neurons, and while some of the data is promising, like preserving the mushroom-shaped dendritic spines in Alzheimer's models and restoring dendritic structures in Huntington's models, not all of it reached statistical significance. It also appeared to delay activation of the MAPK pathway and reduce reactive oxygen species, which hints at potential antioxidant and anti-inflammatory activity. And when we move into animal models like prenatal hyperhomocystinemia in rats, Pinealon showed normalization of central nervous system function. So yeah, there's activity, but again, it's a bit scattered and hard to really hone in on what it could benefit in humans and if this research really even translates. And when we do, it becomes a bit more conflicting. There was a study on 72 patients with TBI, traumatic brain injury, where Pinealon was given orally alongside standard therapy, and there were reported improvements in these different facets like memory, emotional balance, headaches, but this study lacked randomization, and Pinealon was just one part of a larger treatment package, which in and of itself is already baseline, as they called it standard therapy. Another study that came out of Russia on stressed truck drivers used a peptide complex that included Pinealon and found improvements in anxiety and mood, but again, hard to isolate which peptide did what as part of this larger complex. And in typical Khavinson fashion, there are citations that are either misplaced or very, very difficult to even verify. Point being, while Pinealon may hold neurocognitive potential, especially in the context of injury, recovery, or neurodegenerative conditions, without proper trials, it's hard to confidently say it delivers on the hype. Fascinating, yes. Clinically proven, not even close. Does it have the Peptide Buddy seal of approval? No, not really, but nothing does, as you can tell by now, and that's because I'm a YouTuber, not somebody giving out medical advice. And no, my opinion wouldn't change even if you offer me a lot of money to recommend certain sources. That's not a dig at anybody in particular, just the ambiguous, financially motivated peptide community. Now, okay, so mini rant aside, of the two peptides, Epitalon is clearly the more researched, as you've probably gathered by now. And even though we've just dissected it pretty heavily, we're still barely scratching the surface. That said, there's very little evidence suggesting either of these two peptides will turn your sleep around, as Segura alluded to. If one has potential in that department, it's likely Epitalon, which seems to have more of a role in stimulating melatonin synthesis, unconfirmed at this time, but the extent of that effect and its relevance to actual human sleep improvement still basically untouched. I'd argue that melatonin itself remains a far more established regulator of sleep-wake cycles with a much stronger body of evidence supporting its efficacy for sleep onset and circadian rhythm disorders. So yeah, injecting Epitalon to enhance lifespan or improve sleep quality might sound cool, but at this point it's premature. You're basically throwing stuff at the wall and hoping it sticks, metaphorically, injectively speaking. Maybe Tom Segura really is channeling the effects of the experimental peptides. Maybe we got it all wrong and he's straight injecting melatonin, just kidding. Or maybe he's just reaping the well-documented sleep benefits of significant weight loss or testosterone replacement therapy, both of which are far more likely to improve sleep in an aging male than anything we've seen from Pinealon or Epitalon, so there's that. Regardless, I hope you enjoyed the video. I know it was more of an OG video, that deep dive that I myself was yearning for. If you're looking for more ways to support the channel, there's Patreon available. There I make user-requested videos, try to stimulate peptide-focused conversation, and I share research that I find interesting. On top of that, there's a 20-page BPC 157 guide that's made available, as well as access to a growing peptide catalog, retro-themed because it really has to be, and because I suck at coding. It's called the Peptide Codex, and links to all of these will be in the description below. But most importantly, I want to thank you for taking the time to watch this video and supporting the channel. I really do appreciate all of you, and you're asking amazing questions on a regular basis that make me think, that make me review what I've read, and I end up formulating a bunch of videos in response to these questions as is. So keep them coming, keep the comments coming, keep the support coming. I appreciate you more than you'd think. But that's enough for me. Have a great day. Take care, and I'll see you soon.
Cut to the chase, evidence-based. Pull up a chair, let's get this straight. Peptide buddy, he's your peptide buddy.